Let's discuss the steps of bacterial transcription from start to finish. So remember, it starts with the promoter sequence. In bacteria, this promoter sequence consists of a -10 consensus sequence and a -35 consensus sequence. What does that mean? That means that polymerase binds in this region, that polymerase recognizes these consensus sequences and knows it's in the right place. The -10 consensus sequence is also called the Prevno box, okay, after the person who discovered it. Now, this promoter lies upstream of the transcription start site, so that's why it has the negative numbers. The transcription start site is a +1. There is no 0. And anything downstream of the transcription start site is going to be numbered plus. The polymerase in bacteria is this holoenzyme. So it has this core that has these 2 beta subunits, 2 alpha subunits, and 1 omega subunit. And then there's a sigma factor that can bind and dissociate from it. The 2 alpha subunits make sure that this polymerase is assembled correctly. The betas are necessary for binding, and the omega ensures proper assembly. The sigma factor helps us to recognize the promoter. So you have a core, 2 alphas, 2 betas, 1 omega. When the sigma factor binds to the core, you get this holoenzyme. So core enzyme, sigma factor. Sigma factor binds to the core, creates the holoenzyme. It binds to the -10 and -35 consensus sequences. We have our holoenzyme. Its job then is to start to open up the strand. And remember, this only requires RNA polymerase here. We don't have several different RNA polymerases in bacteria like we did in replication. So we pull it apart, we start to add in the first couple nucleotides, and then sigma dissociates. And that's the end of initiation. So core polymerase and sigma bind together and bind to the promoter sequence. Open up the DNA, insert a couple nucleotides, sigma comes off. That's the end of initiation. Next we enter elongation, where our RNA polymerase just goes along, reading the DNA template, creating the RNA strand until it reaches the termination site. There are 2 ways that this can be terminated: a Rho-dependent or a Rho-independent termination, okay? So one requires a protein or enzyme, the other just does not. It requires sequences. So when we look at Rho-dependent, we need 2 sequences here too. We need this rut. So this Rho utilization site, RUT, this rut is where Rho is going to bind to. Rho is a protein that can travel along the RNA strand. So once polymerase synthesizes the rut, so once it transcribes it, now Rho can bind to the rut. When Rho binds to the rut, it rides along it, chasing behind polymerase. So think of this like Pac-Man. So Rho binds to rut, and now it's going after polymerase. So it's going up the DNA, moving. Then there's the terminator sequence on the DNA. Once the polymerase transcribes that terminator sequence, it forms a stem loop, because this terminator sequence has GCs. And remember that in order to form a stem loop or have an area of double-stranded RNA, you need complementary sites. So because this is full of G's and C's in orientation, it allows them to bind together. So once this stem loop is created, it causes polymerase to pause, okay? So it's some kind of conformational change that occurs, and it causes it to pause. When it pauses, Rho is allowed to catch up. Once Rho catches up, and Rho has helicase activity where it's knocking off. You see, if you look here, how it looks like the RNA and the DNA temporarily bind to each other. Rho's job is trying to undo that. It's making these connections unstable. So once it goes along and it catches up to polymerase, it's able to knock it off. And so by dissociating all of those bonds, it makes it unstable. It knocks it off. DNA winds up to how it was. Rho's recycle, polymerase is recycled. You have an RNA molecule that can go on and either be modified if it is the end functional unit, or leave the nucleus in search of a ribosome to be translated. Okay, so this is another way to look at it. You have your polymerase, your rut site, where your Rho binds, chases it along, gets to the stem loop, knocks it off. Okay. The other one is Rho-independent. Now, Rho-independent doesn't require a Rho, hence it's Rho-independent. But it requires nucleotide sequences. So one is a uracil sequence. And then the uracil sequence has a complementary sequence that can cause polymerase. So what happens that causes it to pause? So what happens here is polymerase goes along, gets to an area where it's transcribing these uracils. This is making for an unstable bond, okay? So because this hybrid here is unstable, and we have a stem loop, and again, we're going to pause, this Nus A is a protein on polymerase that promotes it to pause at the stem loop. And when it pauses at the stem loop, it's like everything just falls apart. So if we look at it here, you have these inverted repeats. These inverted repeats are going to be complementary to each other. And then you have the area that codes for the uracils. So after these are transcribed, you get these inverted repeats, you form this stem loop or hairpin, then you transcribe the uracils, and this is causing the destabilization. This is causing polymerase to pause and then to fall off. And then you terminate. Okay? So for the promoter, you have two consensus sequences, a -10 and a -35. Those are upstream of the start site. Then you have the bacterial RNA polymerase that had the core plus sigma equals a holo that binds to that promoter region. During elongation, we just chucking along, filling in the blanks, making complementary base pairing. We get to the end, it can be Rho-dependent or independent. If it's Rho-dependent, it requires a rut site, and it also forms a hair loop. If it's Rho-independent, it's going to look for a hairpin or stem loop as well as a uracil. Okay, so those are the two differences. But the idea is the same, that it causes destabilization between polymerase and the DNA, and once it falls off, everything dissociates and goes back to normal.